Assalamualaikum everyone, welcome back to this channel. This is me Arivaid and you are on the channel of Medicare. So as you know, we have started the chapter of antibodies. So we have discussed the classification of antibodies, its structure, and we have discussed it in a short introduction. Now we are going to discuss some other types of antibodies are related, and after that we also discuss some abnormal antibodies. So how they are produced and what they are doing inside your body. So we will also discuss that. And in the next lecture we will discuss the monoclonal antibodies and what are them. We will discuss the whole lecture on it. So first we will discuss some other types of antibodies, and it is classified on the base of their on the base of their differences in them, like genetic differences or amino acid sequence differencing. So the first type of antibodies is the isotype of antibodies. So these are the isotypes of antibodies, and what are them? Naturally antibodies are divided into five classes: IgA, IgM, IgE, IgD. So these five classes are given, but now these five classes are again subdivided or classified into subclasses, and these are called as isotype. Like if I give you example to you, like IgG, it divided into four subtypes. IgA divided into two subtypes, and other antibodies are also divided, but they are not famous as well. So therefore, these two antibodies are major and they are mostly popular. So what is the difference between them? What is the difference between G1 and G2? What is the difference between G3 and G4? What's the difference? They are belonging to a same group. They are IgG, but why? What kind of variation occur between them? So the variation which occur between it, this variation is called as isotypic variation. Mean these variation make every type of antibody different. How these variations are given below due to amino acid sequences changing out. We have discussed in the previous lecture that antibodies are the protein. So every protein is made up of amino acids. So whenever there is a change in the amino acid sequence, it causes a variation. It causes a change in the antibody. It remains protein, but due to change in the sequence, like if amino acid has to attach at that part, if it changes its equation and attach to that part, it changes out. And also due to genetic mutation, whenever the genes change, everything is going to be changed. So what is the location of variation? Like at what part or at what site these changes occur? So it occurs in the FC region, in the constant region of heavy chain, which also called as the H chain. So one thing you have to remember, I have drawn the structure of an antibody and I have produced some spaces in them. So actually it is not given here. The reasons to make such kind of empty spaces in it is to identify you that the variation is occurring in that part. So as I have explained that in heavy chain variation is going to occur. So this is a heavy chain. Okay, so this is a having chain, and in the FC region, in the constant region, so this is your constant region and this is your variable region of it. So in the constant region, how the variation is occur, I have explained to you with the help of diagram, so that you can clearly memorize it. Now let's discuss the other types of antibodies as well. So this is the next type of antibody, and this is about the idiotype antibody. So what it is these are the types of antibodies which depend on the antigen epitope nature. So you know very well, on the antigen epitope is present. What is the function of epitope? Epitope is the site present on the antigen, and it is the site on which the antibodies are going to attach. And antigen can have one or more epitope on it, and every antigen can have different types of epitope on it. Like we have discussed the types of epitope when we have discussed the lecture of antigen. So we have discussed that epitopes are of various kind. They can be simple, they can be complex, and they can be very complex in nature due to their structural changes. So they can different with each other. So if there is the difference in the epitope of antigen. For every epitope, specific kind of antibodies will produce. Like if I am giving the example to you, consider you have an antigen having two different epitopes on it. So I am making an antigen, this is the antigen of you, and it has two different kind of epitopes. Like if I am changing the shape of it so that you can clearly visualize it, that there is a change in it. So one is in the circular form, other in the square form. So now for this antigen, specific kind of beta cells will produce specific kind of antibody, like it produce IgM against it. But for this epitope, specific kind of beta cells produce, which produce another type of antibody. It could be IgM, okay, it could be IgM but different. Like if it is IgM1, it will be IgM2. It can be anything. It doesn't depend that it always become IgM. It can become IgA, it can become IgG. It depends on the epitope. It depends on the nature of the antigen. If the nature of antigen is that, then the specific kind of antibody will produce. I have just given you here the example. You can write any kind of antibody here. You can write anything here. Okay. So now next move on to other type of variation, where this idiotypic variation occurs. It occurs due to amino acid sequence changing and genetic change. One thing I have forget to tell you, that these antibodies are antigenic determinants, because their type, their subtype is depending on the antigen, so they are antigenic determinant. So what is the location of these variation, where these kind of variation are occurring? Very simpler, it is occurring on the variable region, mean fab region, in both kind of chain, heavy or light chain. So now I have drawn the spaces in that part, in the variable region. It is not the constant region, and I have drawn the spaces in both chains. Okay, as you can see here, so the change occur in the paratope. Paratope is the site of the antibody on which the antigen is attached. So in that part, when the variation is occurred, then for every variation, specific kind of antibody will produce against that. Next topic is about the abnormality of antibodies. Like we can have a lot of abnormalities. Like the abnormal antibodies can be produced, they are cancerous, but their structure is similar to the normal antibodies, but they cause cancer to you. So what types of diseases can caused by the abnormal antibodies? So let's discuss them. The first disease is the Johns protein, can also be called as multiple myoma. Multiple mean very excessive production of anything, and myoma mean any kind of cell. So it is a condition in which neoplastic condition occur, mean increase in the plasma cells. As a result, it is a light chain disease in which the light change of your antibodies will go, and they occur, and there is disease occur in it. There is a very high production of that light chain and causing the production of cancerous plasma cell. They will begin to accumulate in that particular patient, in the patient's serum, and then they will release out through your urine. And they can, these specific kind of antibodies or the plasma cells can coagulate at the 50 degrees centigrade temperature, and they can redissolve at the 70 degree. So sorry, I have written the percentage sign with it. It was my mistake. There is no percentage sign is here for them. There is a centigrade sign should be here for them. Okay, now let's discuss the second disease, which is the Waldenstrom macroglobulinemia. It is a disease in which your lymphatic system disturbs, due to which the beta cells increase their production. As a result, the IgM antibody increase its production. And what is the reason behind it? Due to the mutation, somatic mutation occur in this gene. This is a name of gene, so that's not the compulsory to remember the name of that gene, but if you can remember, then you definitely remember it or memorize it. but if you can't, then leave it, just leave it. So this is a gene name MYD88. So if the mutation occur in that specific gene, you can face this disease. And about 90% of the patient is facing this disease due to this mutation in that gene. Next disease is the heavy chain disease. This is a disease in which there is increase in the production of your heavy chain, not the light chain. The first two diseases which we have discussed, they are changing in the light chain. Now the heavy chain, it caused the excessive production of heavy chains which are short and turnicated. Turnicated mean without Fc region. So if the antibodies are produced without that constant region, your antibodies are not going to work properly. They cannot going to bind properly. So as a result, they will cause the abnormality in your body. They cannot perform the function of protection. They cannot neutralize. They cannot precipitate. They cannot perform all the biological and physiological processes inside your body. Next is the types of heavy chain diseases, having three types: alpha chain diseases, gamma, and delta chain diseases. Next type of disease is the cryoglobulinemia disease. This is a special type of protein due to which this disease is occurring, and this protein names is cryoglobulins. This protein release and this protein cause the IgM to consider IgG as a foreign particle. As a result, IgM destroy all the IgG antibody inside your body. As a result, IgG antibody completely vanish out through your body. As a result, your body will face this disease. So this protein, nature is discussing here that this protein is insoluble at low temperature but soluble at high temperature. So this was all about our today's lecture. I hope that you will understand it. If you like this video, then please subscribe to my channel, like my videos, and also press the bell icon to get the notification of all the videos which I have uploaded. Thank you so much for watching this video for the last end. Make sure that you subscribe it. Meet you soon in the next lecture. Till up, Allah Hafiz. Remember me in the prayers.